What is RT-qPCR? Well, to understand that, you need to know what RNA, DNA, and polymerase are. Think of DNA as a briefcase of instructions for all cells, where genes are the specific steps, while RNA is a person holding the briefcase, taking those instructions or genes to the rest of the cell, so that it knows how to function and make a cell what it is. Then polymerase is the copy machine of all the genes in the briefcase that duplicates the genes it has the materials for. Well, now let's talk about what RT-qPCR is. It's a way of amplifying genes from RNA that are extracted from cells and then quantifying the results, or getting the specific number of those genes present in the RNA. But the tricky thing is that PCR only works with DNA, so how can one determine the genes found in the RNA? Well, that's where the RT part comes in. It stands for reverse transcriptase, which is an enzyme that generates a complementary strand of DNA, or cDNA, from the RNA obtained from the cell. Well, now that you have cDNA, you can do PCR. PCR, or polymerase chain reaction, uses a soup that contains building blocks that are unique to the gene you want to amplify. This allows you to recreate a DNA replicating factory. Thus, the only genes that are being replicated by polymerase are the ones that have the materials to be made. Now, let's talk about the Q. The Q stands for quantitative, which refers to the way we determine the number of genes present in an RNA sample. We do this by placing a fluorescent dye in the buffer that the machine can detect, because it lights up. This allows the machine to count the number of genes present and give us data to analyze. And there we go. If you want to know what genes are being expressed in a cell, use RT-qPCR.